All right. Good evening. Thank you for attending our symposium on cancer evolution across scales. Our first talk this evening is going to be from Jeffrey Townsend from Yale on cancer evolution from mutation rate variation to stage-specific and epistatic effects.
Great. Thank you to the organizers for inviting me to give this talk and to all of you for attending it. I'm going to move quickly, and I hope you all are ready. So first of all, cancer evolution is quite multifaceted. You're going to learn a lot about that during this symposium. I'm going to focus on some of the molecular details, in particular of the many different ways that evolution can happen. I'm going to be focusing on point mutation. Now, mutations of any kind happen in a cellular lineage that then leads to sort of cancer growth and drug-resistant metastatic spread of cancer, typically. That branching process with the mutations appearing is really important to understand, to basically understand how cancer happens. And one of the properties of that mutation process is that it was early on recognized that detection of cancer drivers was compromised by variation in somatic mutation frequencies, meaning the faster mutations happen, the more likely you are to see a mutation in a tumor, and that means that seeing a mutation in a tumor doesn't really tell you how important it is. Like, the more of that mutation you see, if you see a mutation in a particular gene a lot, it may or may not be important. So that was early on recognized, and they pointed out that there's very big differences in the amount of mutations that occur in different cancer types. Along the x-axis, these are cancer types. And the baseline mutation rate varies a lot along cancer types, but it also varies, this is a log scale, a lot within cancer types. So all of this sort of needs to be taken into account to try to understand if that underlying mutation rate is affecting how important a given driver gene is to cancer, what the importance of a given driver gene is to cancer. And also, you have very big differences in very specific nucleotide changes, like C to T, C to A, C to G, T to C, T to A, T to G, et cetera. Color banded here. For different cancer types, you have different distributions of what kinds of changes happen, and that's because of some exogenous factors that I'm going to talk about later in the talk. But the main thing with regard to genes is that mutation rate varies widely across the genome, and it correlates with DNA replication time and expression level. I'm not going to go into detail about this, but you see these lines. These all correspond to replication time, mutation rate, and where across the chromosome is. They're all correlated, and they all relate in a very common way. And so what you can basically know is that expression level and replication time both correlate very strongly with mutation rate. And that means you can use those to predict mutation rate for a given gene. And that was really helpful when people analyzed tumor sequence data, because then they could basically calculate a p-value, which is represented on this x-axis here, for whether or not a gene was a driver or not. And then they could look at that p-value as well as the prevalence of a gene, and then they could know everything they need to know about cancer drivers. Except neither of those things are an estimate of how important it is to cancer. The prevalence is a measure of how many people are affected by that mutation, and the p-value is a measure of belief that it is a driver or not, has nothing to do with the amount of a driver that it is. So to quantify that selection coefficient or scaled selection coefficient, I'm going to call it cancer effect size in this talk. The prevalence that you saw there has to be deconvolved into the baseline mutation rate and the degree of selection for the mutation in the cancer lineage. And that's just because, of course, mutation creates variation. This audience doesn't need to hear this, but unfavorable mutations are selected against. Reproduction and mutation occur. Favorable mutations are more likely to survive, and they're more likely to reproduce, which means both mutation and selection are both affecting the frequency with which you see mutations in tumors. So this mutation rate at each site can be calculated by convolving the gene-based rates from silent sites, as we do typically in evolutionary biology of organisms, and covariates with trinucleotide context. That's something that we don't so much do with organisms. So basically, we can figure out every gene for how likely it is to mute by looking at those covariates I told you about earlier, replication time and expression level, and looking at the synonymous sites that are very infrequent in tumor genomes. So you can't use those directly for every gene, but with those covariates, you can figure out the rates for every gene. And then you can convolve those with these site-by-site rates that I told you about earlier. This is for lung cancer in particular, and you can see all the C to A changes that happen because people smoke. And those can give you a prediction for every single amino acid across all the sites of several genes for what the likely neutral mutation rate for every single site was. And then you can compare those likely neutral rates to the rates we actually observe mutations in the tumor genome. And what you can see is there's a pretty big difference here between what we see in neutral genomes and what we see in the actual tumors. And the reason for that is selection, because selection drives all of these changes in these three genes I'm not going to go through the genes right at the moment because I don't have enough time with the number of slides I'm going to talk about. But I just want to point out that a number of the low-frequency mutations, mutations you don't see so often in tumors, exhibit very high effect size in lung adenocarcinoma. These two highest ones are the ones everyone knows about, KRAS and P53. If you know anything about cancer, those are really frequent genes, and they actually have very high effect. But as you go down here, Actinium B1, the size of the circle here is how prevalent it is. Not very prevalent at all, and yet it's the highest, the next highest selected gene, et cetera, going down. So you can't just take how frequent it is in tumors as an estimate for how strongly selected it is. This is how strongly selected it is. This is the fixation rate, or how frequently you see them in tumors, and this is the mutation rate, which is how frequently the mutations are happening in cells, and they vary extensively across the range over orders of magnitude, which means you really need to calculate those things. The consequence of this kind of calculation, which we do, is that we can then actually estimate the selection coefficient for all of the changes we see in tumors. And so this is a density plot of the selection coefficient, or cancer effect size, scaled selection coefficient, cancer effect size. And what it tells us is that, that there's a distribution. You can see the non-synonymous and synonymous sites here. So these are all neutral ones. And then there's a long tail, which you can't really see in this diagram, of highly selected changes. And we can do this for all the different cancer types. And I'm not going to go through these plots, but the main point that I want to urge you is that if you're doing any kind of simulation or any kind of anything about cancer, do not assume selection coefficients are 0.2 or something like that. We know the answers. So take those answers and use them. Because it's probably the only evolutionary system that I know where we know what the strength of selection, of the distribution of selection coefficients on genes are, because it's repeated so many times that we can get the data out of it. We can get this answer out of it. All right, and these don't mean nothing. They mean a lot, because if you take, so there's this thing called the ClinVar variants, which are clinically validated. These sites are actually really important. And if you look at whether or not clinically, what the scaled selection coefficients of clinically validated ones are versus ones that are not clinically validated, this is clinically validated, not clinically validated, not. In the case of recurrent SNVs or single mutations that we only see once in the cancer data, either way, the cancer effect size is predictive of whether or not it's a ClinVar variant, meaning it's associated with that. Not only that, if you look at polyphen or SIFT, these are scores for predicting whether something has cancer impact, they're not nearly as useful in a multiple regression as the mean cancer effect, which is extremely useful for predicting whether or not something is a ClinVar variant. Not only that, if you do some AI sort of study, I have this amazing graduate student, now professor at URI, Nick Fist, who basically took 21 different data sets, 76 tasks, and 3 different AI approaches, and analyzed all of them. And in every single one, it turns out the cancer effect contains all the information you might want to know. It either does better or just as well as any other combination of these kind of known measures for whether something is maybe a cancer driver mutation or not. Not only that, maybe you're more molecular and you want to see something that really makes you feel like, you know, it's real. Well, this is the gene SPOP in prostate cancer, which is a very, very major driver in prostate cancer. And what I've done is just colored all the residues along the binding portion for the ligand that SPOP binds to. And you can see the selection coefficients, which are down here in this bar graph, are higher for anything closer to that binding and lower for anything farther away. And it aligns perfectly with that. There's big differences among different sites in terms of total selection coefficient. That's a very important observation in its own. But the way they're correlated along this binding groove, you can see them along the groove and correlated with how much they obstruct the binding. It's just a beautiful result. All this just from human tumor sequence data, nothing else. Okay, so cancer effect sizes are this key metric for a full range of basic and translational research. Basic research, which genes should I study to understand the basic biology of cancer? Precision medicine, suppose you're deciding between 2 drugs, one does one thing, one does another thing. They're equivalent in every other way, and you're using them out of, like if they're approved for one cancer, you're looking at a different cancer, and you're trying to decide which one to use on a patient. Use the one that has a higher, that's affecting a mutation that has a higher cancer effect size. Motivating us for clinical trials, extremely expensive. We can only do a few of them. Let's do it for drugs that actually treat mutations that have a high effect size, and pharmaceutical development should also prioritize by this method. Okay, next thing. These mutations, they happen, and they happen in a couple different ways. Sometimes they're clock-like and endogenous processes that are just because of aging, basically, in various ways that we really can't control with exogenous differences. Other ones are caused by exogenous things like viral infection or tobacco smoke, or UV radiation. So we get different mutations at different places in our body and in different tissues at different rates depending on all kinds of different factors. Now the cool thing is someone else, totally not me, figured out that you can take these tumor SNV profiles and you can deconvolve them into known mutational signatures. So you can figure out, oh, was this UVA that caused that mutation, or was it tobacco smoke? It's probabilistic, but it's pretty good in its probabilistic way. So the extent to which these contribute to tumor genesis has been unknown, because we can look at a genome and we see the processes there, but we don't know how much it contributes to cancer unless you use the approach I just talked about to quantify how much it's contributing to cancer. So you've got your different mutations, and we know how much they're contributing to cellular proliferation and survival, but we also know which kinds of processes are most likely to have contributed those mutations, sunlight or aging or something like that. So we can basically go backwards on this little plot and calculate the extent to which every mutation was caused by, say, in this case, tobacco smoke, which is in brown here. And this is in lung adenocarcinoma, lung squamous cell carcinoma. They have different genetic bases. LUAD is mostly caused by oncogenes up here, KRAS and BRAF. LUSK is largely driven by tumor suppressors. And so you can actually map out which kinds of mutations are happening due to which kinds of cancer types. And ultimately, in multiple cancers, you can even then look at what the origin of that cancer is in individual patients. So each one of these is an individual patient. On the left of each pair of bars is the contribution to the genome, and on the right is the contribution to cancer. And sometimes the contribution is equal, and sometimes the contribution is very different. Sometimes it's quite similar, sometimes quite different, sometimes quite similar, sometimes quite different. So, so for 4 different cancers, so we can actually now look post-cancer at someone's tumor and actually determine what caused their cancer, which is super new development I'm excited to make possible for individuals to do and learn more about their cancer if they're interested. So the mutational input from a source is not necessarily proportional to its cancer effect. This is looking at different cancer types. Some of you may have heard about this this question of like, is it bad luck or not? This basically answers this question, at least if you think about it in terms of aging or some exogenous exposure. We have proportions for every different cancer type. You can see things like LGG, which is a brain cancer that happens to people late in life, almost all aging. Skin cancer, almost all UV exposure, so stay out of the sun. All right, and they're different from each other. That's all this diagram says. I'm running out of time, so I'm just going to skip through a few things here. But mutation rate increases with prostate cancer. We can look at how selection changes across oncogenesis. So here's SPOP, which I mentioned earlier, and you can see it's very strongly selected early, not strongly selected in metastases. Here's AR, which responds to therapy, and you can see it's not actually selected early on. It's a response to therapy because you see it selected in metastases taken from patients after they had therapy. We can also look at epistatic selection, and I'm afraid I don't have the time to talk through all this, but we can look at every pairwise interaction between different genes, and we can even look at different genes in different contexts and map it out not just to the mutation rate on the bottom here, but actually look at what the selection coefficients leading to each changes, and this gives us a projection for the future for patients, enabling us to basically understand the evolutionary landscape and where patients are going. And if we look at smokers, you can see they have much higher mutation rate than non-smokers, and that mutation rate is relevant. Not only is it relevant, but it also translates to selection. So here's KEAP1 selected much more strongly in an ever smoker than in an never smoker because it changes the phenotype of the cells in the lung as well as changing the genotype. And that's all I have to talk about today. That's the bottom line messages, which I don't have time to repeat, but thank you very much for your attention. So it's different in every different cancer type? Yeah, and in fact, you could look at subtypes. So if you wanted to look at BRCA, we've looked at this at like ER positive, ER negative breast cancer, et cetera. As you get more and more subtypey, the effects tend to be less different from each other.
Okay, thank you very much to Zach and Diego for allowing me to present today. I will go much slower than Jeff because it's 6 o'clock in the morning in Australia. And I'm severely jet lagged. I'm just waking up, so by the end of the talk, hopefully I will be awake. So let's figure out how we go to the next slide. Button. Okay, I might just go with the... Okay, so again, thank you for Zach and Diego to allow me to present theoretical work. It's rare that we are given the opportunity to talk about something which emerged from somebody else's work and which gave us a lot of joy and a lot of pleasure to come up with some ideas. So recently there have been a couple of papers, seminar papers emerging on transmissible cancers. These papers have been focusing on sequencing the genome of those cancers. And thanks to these papers, we now start understanding the evolution of these cancers, and we can start having some amazing and interesting questions. So this work was actually originated by two of those papers coming out last year, and the journalist asking my opinion about those articles. And I got excited and wrote a whole page about this is my opinion. And then the journalist took one sentence from my whole page. And I thought, bugger it, I already spent a lot of time writing a whole page. Let's flesh out these ideas. So I've started discussing the ideas with Georgina, who was a PhD student in the lab that time, James de Gregory, who is a molecular biologist, and Fred Thomas, who is an evolutionary biologist. And again, these are ideas which are a lot of theories, a lot of questions, and I would like the audience and everybody to start thinking and come up with cool ideas. Before I can go to transmissible cancers, thanks to Jeff, I don't have to tell you too much about the evolution of classical cancers, but just super quickly, cancer is actually a disease of 100 different types of diseases, but they all have one common denominator, is that they originate from one cell, which goes rogue, starts proliferating without control, and start invading other tissues. Now we know that this whole process follows the Darwinian principles, but even if it's following an evolutionary Darwinian discipline, the end of classical cancer is always the same. They always die out with the host. So their fitness disappears as soon as the host dies. If some of these clones are able to jump to another individual in the same species or in another species, their fitness will increase, and they actually become immortal transmissible cancer cell lines. So now we know that there are minimum, and I say minimum 11 transmissible cancers known at the moment to science. And the reason why I'm saying this, because I reached out to the BTN, the bivalve transmissible cancer community, just before I came, and some of them told me that there were 8 of them, some of them told me there were 11, so I go with minimum 8 bivalve transmissible cancer in bivalves. There are 3 of them in mammals, 1 in dogs, and 2 in the Tasmanian devils. Again, thanks to those studies, I jump back a bit. I said there are minimum 11, probably 14 known at the moment, but we have an amazing postdoc, Antoine Dijon, in the lab, who with Georgina, the PhD student, did some amazing mathematical modeling and actually showed that there should be hundreds of these cancers in nature. We just haven't discovered them. We should start looking for them. So thanks to those papers, now we know the approximate age of these cancer cell lines. We know that CTVT is approximately 6,000 years old. The FTD in Tasmanian devils, the older cell line, emerged around 40 years ago in the early 1980s. The younger cell line emerged more recently in the 2010s. And the 2 genome papers, one was on the soft shell clam, MAR-BTN is the abbreviation for that cancer cell line, is minimum 200 years old, but the authors actually reached out and told me that it's approximately 400 years old. And other coccal transmissible cancer is, the authors estimated it to be between centuries or millennia old, so they didn't give an exact estimation. We also know their distribution, or start to recognize and see their distribution. We know that CTVT is distributed across the world, while the Tasmanian devil cancer cell lines are at the moment restricted to Tasmania because the species is restricted to Tasmania. Those 2 one of the cell lines, the older one, spread across the island and wiped out the Tasmanian devils, and the other one just recently emerged in the early 2010s. MAR-BTN is spread across the eastern coast of US and Canada, while the coccal transmissible cancer is spread across Europe. So they have quite large distribution, especially the older cell lines, and those which are not restricted by the island geography. Lots of data in this table. I just pulled out the data from the genome papers. What I would like you to focus on is the estimated mutation rate per year. These numbers were given by the authors. So as you can see, CTVT, DFT, and MAR-BTN have a much higher mutation rate than normal human cancer cells or even somatic cells. So this is where we started thinking with our colleagues that, hang on, these mutation rates are huge. Like MAR-BTN is carrying 6,500 mutations per year. This is an enormous mutation rate for a clonally transmitting, clonally reproducing cell line. How do they survive? How can they do it? And we were asking this question because we know from asexual organisms that when they are reproducing without recombination, mutations, including deleterious mutations, can accumulate, and eventually that will result in the meltdown of those genomes. So again, if I just come back to this image, that we know that CTVT has spread through millions of dogs. The devil cancer is spreading through probably 150,000 Tasmanian devils. How do they survive? They have been clonally reproducing for thousands and thousands of years. How do they survive? And this is where we started putting ideas together. These are, again, just hypotheses, and we started with the classical ones that, well, the simplest way is that there is purifying selection, which removes the cells which carry those deleterious mutations. And then because the number of cells across the host is so huge, that population size will control formulas return. Another idea which we had is that probably these mutations are accumulating in regions which are not essential in the genome. These cancer cell lines have huge genomes, so probably all the mutations are present where they don't matter. And then we know from all of these cancer cell lines that they tend to go through ploidy, tetraploidy. This is an image from the coccal transmissible cancer. And we thought that probably this chromosome duplication might rescue the genome which is accumulating deleterious mutations. All these ideas are the classical ideas which we usually use for asexual organisms, and they all indicate that we have to rescue the genome somehow from the accumulation of mutations. And they all indicate that accumulation of mutation is a negative fitness has a negative fitness impact on the genomes. And this is where we started thinking that, hang on, there are some literature out on human cancers and also on other asexual organisms where actually accumulation of mutations is a positive fitness effect. And there are so-called mutator phenotypes which have a higher fitness than the classical non-mutator phenotypes. And these papers originated in the 1970s when Laurence Loeb proposed a theoretical work saying that excessive accumulation of point mutation is actually beneficial for the asexual organisms, including cancer cells, and it has a positive fitness effect. So this was in the 70s, and then 40 years or 30 years later, he, with another colleague, actually developed a technique to measure that number of random genetic mutations, and with that technique, a sophisticated technique, he actually saw that there are 200-fold higher level of point mutation in cancer cells than in normal cells. So he came up with the mutator phenotype idea and theory that this is actually, having a mutator phenotype is actually beneficial for cancer cells. You are evolutionary biologists, so probably you are all aware of the famous long-term dynamics of adaptation in asexual populations experiments when they have been following 12, I forgot the name, jet lag, doesn't matter. So they have been following the population of 12 asexually reproducing organisms. E. coli, thank you, somebody's telling me, and they actually found that they also have a hypermutator phenotype, which is much more beneficial and have a higher fitness than normal mutators. And there have been other studies also showing that different environmental and different circumstances actually can select for higher mutation rates. One of the studies by Rosenblum, the other one is by James DeGregory, who is a co-author on our article. And they actually came up with the idea that there are several factors like fluctuating environments, strong cyclical competitions, host-pathogen interactions, and competition between different populations can create an environment which will cause the emergence of these mutator phenotypes, and they will have higher fitness. So if I want to translate that idea for transmissible cancers, probably the best example is the bivalve cancers, which are exposed to these really critical harsh environments. As you know, bivalves live in the benthic floor. They are living in intertidal zones where they are exposed to fluctuating water temperatures, and the way these cancer cells are spreading is by mussels and cockles releasing the cells into the seawater. They have to survive in a high salinity, and then the next cockle takes them as filter feeding. So this environment creates this cycling extreme environment which could select for the mutator phenotypes. What about mammalian hosts? Well, they have an adaptive immune system, which the cancer cells have to overcome somehow and hide from it when they are transmitting. And we know also from Rodrigo's work, and he told me he won't talk about it too much, unfortunately, but we know that co-infection of cancer cell types and cancer cell lineages can occur in the host, and they can compete for the host, which creates that competitive environment, which will result in the emergence of mutator phenotypes. Okay, so an idea which came from Fred, who is an amazing theoretician. He came up with the idea that what if this mutator phenotype is actually a compensation for these cell lines being in a continuous red-green disadvantage. We know from previous works that classical cancer emerged at the same time as multicellularity emerged on the planet, and we know from a lot of scientist work that metazoans have evolved really good cancer suppression mechanisms. So there is a long evolutionary history between the host and classical cancers to suppress the emergence of neoplastic clones. So again, metazoans have developed an effective cancer suppression defenses, and transmissible cancers emerged later in evolutionary time. The oldest one, as you could see, was 8,000 years old. So they actually have to combat and work real time with the host immune system and also with that evolutionarily developed host defenses. So to do that, they are in an evolutionary lag phase non-stop with the host, and we propose that this high mutation rate and mutator phenotype may be to overcome this red-green disadvantage. Okay, a couple of questions we have been discussing in the paper and we don't have answers, and I sat through a mitochondrial session yesterday and I was excited to hear about mitochondrial nuclear genome interactions. We know from some of these transmissible cancers that they capture the host mitochondria to rescue their own mitochondria, and that mitochondria can take over in the cells. If they do that, they will break down the cooperation between the nuclear and mitochondrial genomes. And would that contribute to high mutation rate? Would that new mitochondria and mitochondrial genome present in the cells would actually boost the evolution and the mutation rates of the nuclear genomes? Another couple of ideas which we were excited about it in human cancer. We know that cancer cells can spontaneously fuse and recombine. And we also know that horizontal gene transfer can occur via extracellular vesicles. So would this be the mechanism saving these genomes from meltdowns in transmissible cancers? Another exciting angle, which hopefully gives you some ideas to think about, bivalve transmissible cancers called jump species. How do they do that? Do they cooperate as we see in human cancers or not? So hopefully I managed to convince you that transmissible cancers are an amazing evolutionary process, and hopefully I managed to convince you that these mutator phenotypes may be a positive outcome, and they may have positive fitness effect, they may not to be rescued. Final slide. If you want to read more, please read the article. There were a lot of ideas I skipped through. because we had a really good time just dumping ideas down on paper. We had 2 amazing editors and anonymous reviewers who helped us to focus, because we had too much fun. And the final slide is thanks to all the funders. And if you want to know about trivial facial tumor diseases, please watch the talks by the students, Georgina, Nick, and Annalisa. Thank you very much.
We have time for one question. Right here.
At what point is it a new species?
Good question. We started calling it, a question was at what point is a new species? We started calling it a new pathogen. I have to admit it has been for a long, long time we debated it, but they are just evolving so weird and so cool that we started calling them a new pathogen.
Very similar to social parasites and ants.
Oh, it's a good point. Yeah, that's a really good point.
Thank you.
Thank you.
Hi, my name's Sophie. I'm a PhD student at the University of Galway in Ireland, and today I'm going to be presenting my work, which is looking at the role of gene copy number in adaptation to life history traits across mammals. And the life history traits that I'm interested in are body size, lifespan, and cancer prevalence. So in mammals, cancer is not seen to correlate with body size or lifespan like we expect that it should. And I say that we expect that it should because cancer can occur randomly in a cell. So mammals that are larger and have more cells should be more at risk of getting cancer simply because they have more cells. So the chance of something going wrong in one of them is higher. And mammals that live for a long time and have a longer lifespan should have a higher risk of cancer because they have more time for these mutations to accumulate. But actually that's not what we see, and we don't see a correlation between body size and lifespan and cancer prevalence across mammals. And this observation is called Peto's paradox. And what this tells us is that large and long-lived mammals have evolved some really effective mechanisms for preventing cancer. And one of these mechanisms that have been suggested is the role of gene copy number. And one of the most famous examples of this is in the elephant. So elephants are really large and they live for a really long time, but they have a surprisingly low rate of cancer. And so researchers have found that in the elephant genome, they have an increased copy number of certain tumour suppressor genes, for example, TP53. And what this means is that they have this redundancy in copies. So if they acquire a mutation in one copy, they have backup copies, and it means that they would require more mutations to trigger a malignancy. So if one copy of TP53 is knocked out by a mutation, then they have more copies, and each of those copies would need to acquire a mutation too in order to trigger the malignancy. So elephants have been found to have around 20 copies of TP53 in their genome. Some of these are pseudogenes, so we're not exactly sure the role that they play, but there has been established an association between the copy number of tumour suppressor genes and cancer prevalence in the elephant. And elephants aren't the only mammal that have surprisingly low rates of cancer. The naked mole rat, for example, it's not very big, admittedly, but it lives for a really long time. So it lives for around 30 years, which based on their body size is like 5 times longer than what we would expect to see. And they have an extraordinarily low rate of cancer. There's only been a handful of recorded cases in the naked mole rat. And so a study that estimated the copy number of all cancer-related genes in the genomes of a bunch of different mammals found that in the naked mole rat genome, they have an increased copy number of these cancer genes. And so when I say cancer genes, I mean any gene that's implicated in cancer, so tumour suppressor genes, oncogenes, etc. And this is consistent with the low rates of cancer in this species. And the idea here is that having this increased copy number of these genes enhances the robustness of their cancer suppression pathways. So this is something that I think is really interesting, and so this project looks into the idea of gene copy number as an evolutionary mechanism to lower rates of cancer in mammals. And I really wanted to contribute some insight into this question, which is, does gene copy number play a role in the evolution of low rates of cancer in mammals? So I started out by looking at the association between gene copy number and life history traits, body size, lifespan, and cancer. It's kind of a similar idea to some of the studies that I've just mentioned that looked at the association between copy number of cancer genes. But instead of just looking at the copy number of cancer-related genes, I estimated the copy number of all protein-coding genes in the genomes of around 100 mammals. And of these copies, I am only counting protein-coding copies as a copy in the copy number count. So again, studies that have looked at this association before have included pseudogenes in their gene count, whereas I'm not doing that. So to look at this association, I used phylogenetic generalized least squares, or PGLS, to see if there was an association between the life history traits and gene copy number. And so we found 15 orthogroups that were associated with rate of malignancy. So an orthogroup encompasses a gene and all of its orthologous copies, both within and between a species. And rate of malignancy is a cancer phenotype. So the data that we use in this study is obtained from published data of another study of zoo animal necropsies. And if cancer was identified, it was determined whether it was malignant or not. And so this rate of malignancy is this metric of malignant to non-malignant cancers. And we didn't find any association between gene copy number or lifespan or body size here. And so I won't go through all 15 orthogroups because we'll be here all day. But some genes of interest to highlight are NCOA4, which is a nuclear receptor coactivator that's involved in ferroptosis. Ferroptosis is cell death that results from abnormal iron accumulation. And cancer cells have abnormal iron metabolism, and so ferroptosis can act as a type of tumour suppression by killing cancer cells. Also TUSC3, which is a candidate tumour suppressor gene, and NEC3, which plays a role in cell cycle progression and cancer progression. So from here, we expanded on our analysis by looking at gene sets. And this is where the study gets a bit different from traditional studies of gene copy number. Because when you're looking at the copy number of individual genes, when you're comparing between species, there isn't actually that much variation. And so it's hard to detect any association, especially when you're looking at so many genes because we don't have much power. So we decided to adapt our methods to do a gene set analysis, where instead of looking at the association between an individual gene's copy number and life history traits, we looked at the total gene count of genes within a set. So for a gene set, so a group of genes that are involved in the same process or pathway, we aggregated the total copy number of all the gene counts in that gene set, and then looked to see if that gene set had expanded or contracted in association with body size, lifespan, or cancer prevalence. And so we applied a couple of different gene set analysis methods. The first one that we did was a PGLS analysis. So the same methods that we're using for looking at association with an individual gene, but instead of using individual gene copy number, we were using the aggregated gene count. And we found 10 different gene sets that were associated with rate of malignancy. So the same cancer phenotype that we were just looking at. So here are those gene sets. And something to note about these gene sets is that most of them have a negative association. So the expansion of these gene sets is associated with the decrease in malignancy across mammals. And that's the case for all of these gene sets, apart from the 2 that are marked with the asterisks that have a positive association. Something that's also interesting about these gene sets is quite a few of them are immune-related, which is interesting in the context of immune evasion in cancer. So on this plot, we have the p-values on the x-axis of the results from our PGLS. And so the gene set with the strongest statistical evidence of association was this top gene set. And this is the gene set I want to talk about a little bit more, which is negative regulation of transforming growth factor beta production. So transforming growth factor beta, or TGF-beta, is a cytokine, so it's a protein that's involved in immune function and cell survival. But it also plays a role in tumorigenesis. So At the onset of malignancy, TGF-beta can act as a tumour suppressor and has been identified as a potential target for cancer therapy, which is interesting because we found that increased gene count in this pathway is associated with a low malignancy rate across mammals, which potentially suggests the role of this pathway and that its expansion might be involved in malignancy prevention in mammals. And so we can actually visualise how copy number of this set has evolved in comparison to rate of malignancy. And we can reconstruct ancestral traits to see how they've co-evolved. So this figure shows the change in malignancy rate across the mammalian phylogeny on the one side and gene count for the TGF-beta set on the other side. And when you put these two together, you can see how both traits change in comparison, which can give you an insight of the evolution of this gene set and malignancy rate across mammals. So when we were investigating gene sets, we didn't just do a PGLS analysis, we also employed some more traditional gene set analysis methods. So we did a gene set enrichment analysis and also an overrepresentation analysis. So both of these methods relied on our individual gene results. So that initial association test we did that looked at the association between individual gene copy number and our traits, we were using those results, we were able to rank genes. And using this ranked list of genes, we were able to see if any gene sets were enriched at the top of this list to do a gene set enrichment analysis. And then for an overrepresentation analysis, we were able to take all of those genes that had a significance of less than 0.05 and use this subset of genes to see if any gene sets were overrepresented in this group. And so by doing these three different methods, we're able to compare our results. And we found 3 gene sets that were statistically significant across all 3 methods. So the first gene set is the one that we've already talked about, which is the negative regulation of transforming growth factor beta production. But then the other 2 are both immune-related gene sets. So they're TAP complex binding and MHC class 1 peptide loading complex. And so our gene set analysis has told us that the number of genes and their copies in these sets are associated with the rate of malignancy in these mammalian species. So expansion of these sets might be involved in some way in malignancy prevention. So just to conclude, I guess my take-home message from this presentation is that gene copy number can be used to identify genes, but it can also be used to identify processes and pathways that might be involved in the evolution of cancer resistance in mammalian species. And our study has identified a number of genes and gene sets where copy number is associated with rate of malignancy. For example, the TGF-beta pathway, where the increase in copy number or the expansion of the gene set is associated with a decrease in malignancy rate, which suggests a role for this process in cancer prevention. And finally, I just want to say thank you to both of the labs that I was working in to do this work, so the Shoiga lab at University of Galway and the Tollis lab at NAU. Thank you for listening.
We have time for 2 questions.
Really nice, looks of your amazing work.
Thank you.
I think in the last slide you showed TEP and MHC being involved, but those 2 gene complexes co-evolved. Because without TEP you won't have MHC on the side surface. So we consider that some of these genes are co-evolving because of the roles and other functions.
Yeah, so there's actually some overlap in some of the genes within the gene sets. So with some of those immune gene sets, there is an overlap in the genes that are involved in those pathways. So I think almost when you do look at those processes, you kind of have to lump them together in some way and say this whole process might be involved rather than these 2 separate entities because there is overlap between the sets.